Hello everyone, this is Syed Muktadir Al Sium from Bangladesh Agricultural University. Welcome to my presentation on AntiFam 6.0. Are we close to getting rid of spurious proteins in sequence databases? Let me start with some facts. As genome sequencing is becoming more common in today's world, many false, that is spurious genes are being predicted during protein sequence prediction using computational methods. Even if we presume the false positive rate of the methods is only 1%, then within a database of 180 million proteins like UniProt, we would still expect to find 1.8 million spurious protein predictions. Now, what are the consequences of these spurious proteins? Future genome projects will annotate new protein coding genes by similarity to the first spurious open reading frame. This can lead to entire families of spurious open reading frames. In the worst case scenario, these spurious families may even be annotated as having a function. For example, a spuriously predicted gene on the opposite strand of ribosomal RNA had been given the incorrect function of cell wall hydrolase. One existing tool that can aid in finding gene misprediction is the AntiFam database. AntiFam is a collection of profile hidden Markov models that can be used to identify members of potentially spurious protein families. However, till now the AntiFam approach does not scale well because identifying spurious protein families is a very slow and manual process, and each family requires the effort of a curator to build it and verify its status as spurious. Thus, only 72 families could be added after the fifth release of AntiFam in almost 8 long years. Another machine learning based tool, the Spurious, was developed to help identify spurious protein predictions in only prokaryotes. The most informative feature for this is the appearance of stop codons within the presumed translation of homologous DNA sequences. Spurious has dependency on AntiFam for training dataset, but actually, as a machine learning based tool, Spurious didn't get a good number of training data from AntiFam because it doesn't have that much. So Spurious is still in its early stage of development, and Spurious results can be considered as indicative, but they require inspection for most applications. The identification of spurious gene is an area of genomic annotation that has received very little attention. This is partly due to the difficulty of proving that a gene is not expressed in any condition. This present study is an attempt to explore those possibilities to find some ways of enriching AntiFam entries with confidence and enabling future improvement of Spurious. For this, we have started searching spurious proteins with Spurious 1.1. Even after having a high spurious score, it seems too difficult to be sure about that protein's spurious status. So we had to find out another way for becoming more confident with our decision. Then we came with BlastX method, and it if you look at this figure 1, at first we take a query protein, and there is no way to do a protein versus protein search in different DNA reading frames. We had to take the DNA version of that query protein, and the BlastX search was carried out, and against it was against the real protein database SwissProt. So what happens if any matches is found a true protein in -1, -2, or -3 frame, this 3 frame, and this is a true protein, and there is a -1 frame. So this makes our protein, this one, in +1 frame, a fake protein, that is spurious proteins. So this was very simple but very effective method, but eventually the idea of using usearch came on our mind because of its swiftness and advantage over BlastX in finding the overlaps. And this method has reduced the time and labor, but most importantly, it gave us the confidence in decision making of protein spurious status. So this approach was much faster, and we could add 164 new families of spurious proteins from the shadow orbs within only few days. This approach was like it can be used as a new separate quality control system for genome annotations, and it will be a strong candidate for being added as a new feature for Spirio to give its prediction much more confidence as well. And if we look at figure 2, it is clearly seen that AntiFam 6 has almost 3.5 times higher uh, entries, and it can find 3 times higher spurious proteins in tremble. So additionally, low complexity regions and protein disorder found in spurious protein sequences had made us interested to have a closer look on this, and IUPRED-2 was used to analyze the protein disorders, and it gives a score for each amino acid of the protein where 0 denotes the no disorder and 1 denotes highly disorder. And if we look at this picture, we created a histogram with the average IUPRED value of the real protein and spurious proteins. We can understand very easily that the sequence having IUPRED average score greater than 0.45 here. So this part has higher chance of being spurious proteins. And so we will suggest that Spirio can be trained with the large number of spurious proteins using our new AntiFam entries, and we can suggest two new features, that is the blast test for research analysis and the protein sequence disorder analysis. We can expect that these steps will play a vital role in the development of a powerful tool for identifying spurious proteins in a greater extent and making the protein databases more purified by just vanishing all the mispredictions in near future. Thank you so much.